Our next speaker is Meaghan Parker. Ms. Parker completed her undergraduate studies at the University of California, Santa Cruz, and her master's at San Francisco State. Her education and career have focused on applying genetic methods to address questions in conservation and biodiversity. She's now a science officer at the Forest Legality Initiative at the World Resources Institute, where she leads the strategic plan at the intersection of academic, public, and private sectors on how to incorporate innovative technologies to combat illegal logging. Let's welcome Meaghan Parker to Duke.
I don't know. Let's see. All right. First, I just want to say thanks. I'm super honored to be here. I'm excited to talk with all of you about all of your great ideas and hope to talk with our mentor colleagues and see how their ideas form together, and we can walk out of here in 24 hours with some really exciting results. Yeah, I just want to say again thanks to the Blueprint crowd that invited me here, but also I know how hard it is to put a workshop together, so I appreciate your efforts and your expediency in email replies. So Blueprint wanted me just to give a little bit of a background of kind of how I got into conservation, so I'm not going to spend too much time on this, but we'll go way back. I grew up in Northern California with my head stuck in the tide pools, counting nudibranchs and brittle stars and mussels, and then went on to be a docent at Long Marine Lab at Santa Cruz, wanted to stay in Santa Cruz because if you know Santa Cruz, it's really amazing, and worked under Dr. Bernie LaBeouf, who was a very famous animal behavioral scientist, and spent time out on Año Nuevo Island for a couple seasons doing animal behavior, and out there I met one of Dr. LaBeouf's postdocs who was kind of on the forefront of using genetic analysis to answer questions in conservation, and she really we had a lot of long nights with maybe a little bit of alcohol on the island by yourself, smelling like bird poop, and a lot of elephant seal, fights going on in the background. But she really keyed me in on how powerful genetic tools can be in answering questions in conservation. So I kind of threw myself into that and went to work with Dr. Michael Soule, who's kind of the father of conservation biology, and he had a couple postdocs working on genetic techniques to answer questions in conservation. So I just continued to go down that path and was really fortunate. Ended up in Hawaii working on Pacific endemic land snails and molecular evolution. And when I was there, I got I kind of got asked if I'd like to head to the Smithsonian and work in Moorea, and that was one of those things where it was like, pinch me. So I ran to Washington, D.C. as fast as I could, and I was the lead molecular biologist on the Moorea Biocode project, and this was a really interesting project. It was $6.5 million from the Gordon and Betty Moore Foundation to basically inventory all marine and terrestrial species on the island of Moorea, and it was kind of a proof of concept. Can we do this? Can we inventory everything on this island in a way that can, make long-term contributions to answer questions in conservation? And it was a massive undertaking. It was 3 years. We had 4 taxa teams. We had 3 different universities and 1 million visiting scientists, and we'd spend, 24/7 in the lab after we would collect underwater, and, it was just hours and hours and hours, hundreds of hours of time in the lab and underwater and on the ground. But in the end, we collected roughly close to 50,000 specimens, and of those 50,000 specimens, about, I think, if I'm not mistaken, it was 5,000 species had not been named before, so pretty exciting, but obviously long-term contributions to efforts in conservation. And I can say that I sampled genetically all 40,000. Maybe I had 5,000 worth of help, but mostly myself. But that was really exciting, but it was more of an inventory. And so I continued on at the Smithsonian, and I was part of this Barcode of Wildlife project, which I thought was really exciting and really amazing. It was a Google Global Impact Award, and it was more applied, and that's what I was really excited about, was that, we could use this really powerful technology, but take it into the arena of conservation and wildlife enforcement. So we could work with the scientists on the ground in country where species, that are of significant trafficking trade leaving and entering the country, and people on the port authorities, enforcement officials have no idea how to identify these species because they're tricky. These guys, these traffickers are not stupid, so they pluck the feathers off, endangered birds so that they're not identifiable. They grind up all the medicinal plants into powder, so even a fairly good or a really good taxonomist is going to have a difficult time telling these species apart. And so you can imagine that somebody who's on the port has no way to be able to identify this stuff. So this was a really exciting project because we were able to go to Kenya and to South Africa and to Mexico and work with the labs on the ground to help build reference databases, genetic reference databases of species of interest that were coming in and out of the country illegally, and set up these networks where you're working with local police, local enforcement officials, port authorities, so that when the port official, assuming he's interested and excited about this project, and we'll get to that later, wants to potentially confiscate a material, he can send, because we've got this great reference database that we set up of species of interest, he can send a sample to the lab, DNA barcode the sample at the lab, and in 2 to 3 days have an answer for, potentially some kind of conviction, So this is really exciting, really applied, and then from there, maybe I was in DC too long, I wanted to go into policy, so I went a little bit more applied, and I ended up at the World Resources Institute, where now I work on illegal logging issues. But what I learned from all of my time at the Smithsonian and prior to that is that the key is really to understanding the appropriateness of the tool, and because you're all here thinking about this, and maybe this is new to you, and maybe this is not new to you, that what happens is gadgets are really fun and they're really exciting, they're bright, shiny objects, but sometimes these tools are built without really a question in mind. So it's thinking about what the questions are and the context around the questions and applying a technology with a question in mind. And something that was new to me was, wow, this is so cool. My genetic, my genetic grounding and foundation and everything I think about genetics, that it can solve every problem. When I finally got into the applied world, it was like, oh yeah, I guess political will matters and legislation matters and governance matters and all these things that there wasn't a context for me. And now I feel like in the applied space, I understand the context, and that is really profound when you're trying to develop tools. And what you recognize is there's not one silver bullet, and I know other people will talk about that as well. So this was interesting for me because coming from the Smithsonian, where I worked on flora and fauna for years and years and years, I didn't really have any idea that there was this huge illegal logging problem. And the reality is, monetarily, in terms of trafficked material in the world, illegal logging trumps every other highly trafficked trade. So, and there's a lot of attention that's being geared toward this now since the 2008 Lacey Amendment, which included plant material. So now this is a big deal, right? Lacey legislation says you have to identify species and origin of all plant material coming into the country, which is great. But what happens is it's different than the wildlife trade. As you can imagine, I'm going to show you just a bunch of animations at this point because they're much more fun than me talking. The supply chain with wood is just tremendously complex. And so you have loggers that are taking trees that potentially are legal, mixing them with trees that are not legal, and mixing that all together. It all goes to China. It comes into Home Depot as furniture for you that's maybe made up of 6 different species. You've got a veneer that's one species. You've got MDF. And telling what species those are is almost impossible. And if it's impossible for a wood scientist to do it from a product that's, at Home Depot, you can imagine when a shipment of wood comes in, not even wood, a shipment of chairs, teak chairs come in for, patio furniture, that the enforcement official just has to look at the documentation and go, okay, it says it's plantation teak, so it should be okay. I just have to kind of take their word for it, and that's the best they can do. So there's a lot of technologies out there right now, and the next set of slides is going to be a bunch of different technologies that we're doing in a spatial way, in a geospatial way, and a non-geospatial way. And this slide is just hard for me to look at because I feel like I came from this world of illicit wildlife trade and working in that, but not really recognizing that even if we stop all poaching, if we don't have forests, we don't have habitat. And I was in Madagascar a couple of years ago, and there's nothing left, and it's heartbreaking, and you realize that because monetarily it's such a valuable trade, and it's so hard to whittle apart because the supply chain is so complex that it's going to take a number of different tools to really break this down and make a big impact, but we have to do it. It's that important. Okay, so here's all of our animation slides. I'm just throwing out, this is for all the students because I want you to really gear your brains around what types of tools could be useful, because there's a whole load of tools, and I'm just going to show you a handful. So we talk about perimeter defense. So what could we do to protect a natural forest? So, we could use something like sniffer dogs. That's something that is, has been used and is being used at University of Washington and then the Working Dogs for Conservation. Sniffer dogs have an incredible sense of smell. They can go to a forest and figure out if there's been, a human camping out there and where, and that could help an enforcement official kind of hone in on where to find a potential illegal logger. There's some really great technology using those old flip phones. Cell phone technology can identify sounds in the forest and say there's a motorcycle or there's a chainsaw over here, and engage an enforcement official at headquarters to get over there as soon as possible to see what's going on. And Rainforest Connection has been leading that work. That's pretty exciting. And these are all partners that WRI has been working with. This is our own Global Forest Watch using satellites to look at forest degradation and forest cover loss. And really fascinating, but it's not real time, it's near real time, right? So you can't, it's great. I think what it does is and the goal here is to shine a light on the countries that are having the most issues of illegal logging and forest degradation and forest destruction. So it's kind of this radical transparency, that makes countries kind of obvious where you're having trouble, and it allows kind of the civil society organizations to call these countries out and the diplomats out and say, look, you have a real problem here that you have to deal with. There's another application, which there's lots of handheld applications, lots of phone applications. GFW just put up another one, which is this little, engaging civil scientists or engaging civil folks on the ground just to really engage in their community, because these forests are livelihoods for millions and millions of people. So if you can engage local communities on the ground to take pictures of illegality that's taking place, and then either alert a civil society organization or alert enforcement officers or something, but just to kind of help engage the people that are really being affected by this illegal logging. So that's all the geospatial stuff, but what I work on, and I'm the science officer for the Wood ID program specifically, is wood identification. So say, you have the satellite data and the trees are now gone, and you can shine a light on the countries that are doing all these illegal logging operations, but at the same time, the wood is now gone, it's off to China, and it's processed, and now it's coming into our country. So even the best wood scientists can't tell these two species apart. It's a Dalbergia nigra from the Brazilian rainforest and Dalbergia spruciana. And one is a CITES Appendix 1 listed, which means it's not allowed in trade at all, and one is the CITES Appendix 2, which means it's allowed in trade but with a CITES permit. And if a wood scientist can't tell these two species apart, bet your bottom dollar an enforcement official can't. And again, they have to just rely on the declaration saying, well, they say it's spruciana, I just got to let it go, unless there's something that's flagging that declaration for some reason. So here, I'm just going to walk through a few of the things that we're working on for the non-geospatial stuff in terms of just wood identification when you're on the port. We've been working with the Forest Service pretty closely and the Fish and Wildlife Service. So one of the things we're looking at, so a decades-old science is wood anatomy. Looking at the cellular structure of wood, you're able to identify different genus of trees, different genera of trees. You're not really able to get down to species, but what we've done, so a xylaria, if you don't know, is just like a curated body of wood, just like you'd have an herbarium, you have it for wood, and it's a xylarium. So we've gone all over the world to take samples of all the different woods in the xylaria. People that are wood anatomists take images on slides and look at the cellular structure in a microscopic way. And so we've taken photos of all of the microscopic slides that exist in the xylaria around the world, and using kind of it's akin to facial recognition software, put all this stuff in a database and used an algorithm that basically uses that same kind of profile where it picks up subtleties that the human eye couldn't pick up. So, and we've been able to do this with the records so far with about 97% accuracy, so it's pretty exciting. But you can see that, or you can assess probably that within the cellular structure of wood, there's probably lots of things that reside, the phytochemicals, the DNA, and the isotopes. So there's lots of different ways to try to figure out either the origin or the species of the wood. So DNA, of course, being a geneticist, this is kind of the end-all be-all, and you think to yourself, we could figure out species and origin, it'd be so easy. And that's great, minus some caveats, right? So again, there's no silver bullet, and this would be the silver bullet if you could really get DNA out of wood all of the time. And in some instances, it's a perfect tool, and in other instances, it's not. So last year, these guys got convicted. There was the U.S. Forest Service knew that something was up. There was illegal logging happening in one of the Pacific Northwest forests, and they thought, we've got to be able to catch these guys, but we don't know how. And so WRI funded this study where we built reference data, taking leaf tissue from the local forest in the Pacific Northwest of big leaf maples, and then, we took samples from the stumps that were logged from the big leaf maples and took samples from the local mill, and guess what? They matched. And that's perfect. That's a perfect use case for DNA, but it's a very boutique use case. So when you're talking about that crazy supply chain that goes through China, this is not a really applicable technology. But what you can use in something like, a really complex supply chain is the chemical analysis, because those phytochemicals don't go anywhere. I should back up a little bit. So where the caveat is with DNA is that, it's a perfect use case for fresh wood, but what we see coming through our ports is heat-treated, chemically treated woods, like flooring, that there is very little DNA, and if there is any DNA, it's highly degraded and probably not useful for genetic analysis. But coming back to this slide, what we do have is the phytochemical analysis, or the chemical analysis, which each species has its own chemical fingerprint. So it's really unique, and it's really wonderful, and it's an easy tool to use. It doesn't take much. It's about 2 cents a sample, and it takes about 3 seconds to process this. And when somebody can look at this on the port, what we're trying to do right now is set up a database for the port authorities so that they can just look at these two fingerprints together. One that's a reference database next to an unknown that they get across, that they get from a seized sample. So a chemical analysis is a really great way to do it. But again, the caveat here is with the chemical analysis, at this point, we can only get to species. So we're still left with, well, maybe it's a species from a plantation that is totally legal because we don't have an origin. But you can do origin with isotope analysis. So because isotopes are, very specific to where they are on Earth, that the tree takes that up, and the isotopic analysis is really indicative of where the tree came from or harvested. So those are just a bunch of different, I'm just trying to throw out as many different tools that we use to kind of stimulate your brains for the next 24 hours so that we can think about tools and in order to deal with illegal logging and deforestation specifically, I guess, if I want to help myself out here and I need help. So. Remaining challenges, just building reference databases. I think that's true for everything. I mean, even what Scott was talking about, the more data just means the science is more robust, right? So we need as much data as possible. Commercial lab capacity, we need to move this out of academia, which luckily this is happening, which is really exciting. Pilot testing, if you can do a good pilot test, you can get more funding. Your funders like to see that things can be scaled, and your pilot, like the Moorea Biocode project, was a great pilot to say that this can actually be done. And then scaling up, we've got to get the private sector involved. We've got to get the private sector to understand why it's important to uptake this technology. So, and I guess my last slide would just be partnerships are key. That's it. You can't do anything by yourself. You really have to work together collaboratively in an open framework to make things happen. So.